Welcome back to the course Fundamentals of General Immunology. After looking into the types of antigen and how these are processed through MHC1 and 2 pathways, how these antigens are important when it comes to immune response, what are the different ways there is a variation in immunoglobulin chains which are possible by VDJ recombination, stuff like that. We will move on to our module number 4, which is immune cell programming and developmental decisions. And in that we will start with lineage commitment of the immune cell, and also what we know about the functional plasticity. So in this module we'll try to learn how these immune cells are programmed, say how they are differentiating into different cell types, and what are the things which are deciding the fate of a cell, so that we can understand the immune regulation in a proper way. So let's start and look into this lineage commitment term in general. So when people are saying differentiation stands for when a particular cell type is converted into its functional unit, or proper cells which are actually functionally viable. So differentiation is a very important phenomena that is governed by multiple factors which we'll study in this lecture. But let's, for example, if we try to see stem cells, so stem cells are actually cell type which are having a potential to form any other cell types. And in pluripotent cells which are having a capacity to produce a limited number of subtypes, but in stem cell we can generate multiple different types. So let's try to understand this term which we call a differentiation of hematopoietic stem cells. Now when it comes to hematopoietic, hemato means blood and poietic means generation. So the birth cell generation, stem cell is just a mother cell. So we know that these hematopoietic stem cells are actually the bone marrow derived cells which are present in the bone marrow. So this is actually long bone stuff, and in the shaft of this long bone we have bone marrow. And we could isolate some of the cells which are called hematopoietic stem cells, which are having a capacity to differentiate into multiple types. So say if we have a hematopoietic stem cell, the two most progenitor cell types which are coming out is called myeloid, and the second is called lymphoid. Myeloid progenitor can give rise to multiple different cell types such as megakaryocyte, eosinophil, basophil, erythrocyte, monocyte, and neutrophils. Now monocyte can again be differentiated into dendritic cells and macrophages, which we have understood very thoroughly in antigen presenting cells, and also this platelet which are coming from megakaryocyte. Now platelets are specific cell types which are responsible for blood clotting, and these megakaryocytes are actually the mother cell which can form the platelets later on. While the lymphoid progenitor cells can be differentiated into T cell, B cell, and the natural killer cell. So the lymphoid progenitor cell here can be differentiated into different lineages which can make cells of the immune system in a different way. So there could be T cell, B cell, which are lymphocytic population, and then we have a natural killer cell which is cells of the innate immune system, while B cell can further be differentiated into plasma cell, which is a memory cell. And these all cell lineages can distribute in the body system in a different proportion. Now any deviation in this cell population can lead to variation in the blood mass, and accordingly we can see deviation in other physiological aspects of the human being. For example, if there is a less of RBC, we can see anemia is coming out. If there is a problem in the platelet, we can see there is a clotting defect. If we have some problem on the lymphocyte, we can see the immune response is modulated in a great sense, and therefore this differentiation is very, very critical in sense of the immune regulation. So let's move on to a separate example, and there we can see a different subset, not a hematopoietic stem cell, but these are mesenchymal stem cell. Differentiation is same. The pattern will be differently regulating a different cell mass. So suppose this is a mesenchymal stem cell. This having a capacity to self-renewal, and also this can be differentiated into multiple cell types. For example, myocyte, which can form the muscle, adipocyte, which are essentially fat-driven molecules or fat-driven cells, osteoblast, which are related to bone formation, chondrocyte are related to cartilage formation, and neurons are the part of central nervous system. So essentially these all cell types are actually forming one way or other way a very separate sets of organ. For example, the osteoblast is actually related to complete bone formation, and of course there are other cell types which can dissolve the cell mass, which are called osteoclast, which we'll see little later. And also in chondrocytes, we see the cartilage functionality will depend upon this cell type. Again, any deviation in mesenchymal stem cell differentiation can lead to altered developmental defects, altered physiological aspects of a human being. Now, to understand the differentiation, we need to see what do you mean by lineages in particular. When we talk about lineage, what does it mean in terms of differentiation? So let's see this immune cell lineages commitment term. So it's an irreversible process. So once the process starts, it is irreversible. We can't go back. So some of the cells which are formed as in RBC or WBC, it's going to revert back to the stem cell. Of course, there are ways to do it in a different way, but generally this process is irreversible. So, and then again, you see different cell types are coming out using this. We already looked into that myeloid cell can form these cells of the immune system, and the lymphoid cell can form again the cells of the immune system, but the different lineages such as T cell, B cell, and natural killer cell. Now these processes are, as I said, these are highly regulated processes, and most of the processes will understand in terms of central dogma. So we know that DNA is converted into RNA, and RNA can convert itself into a protein when we are talking about a messenger RNA, for example. So now this process is what we call transcription, and this process is what called translation. So translation phenomena and the transcription phenomena are controlling these two. So DNA, so DNA to RNA conversion we call transcription, and the mRNA to protein conversion we call translation. Now DNA to mRNA conversion, there are a lot of proteins which are responsible for converting this, and those are called transcription factors. There are regulators which can control this synthesis of RNA over the DNA template, and these are regulated nicely in order to facilitate the production of a protein in later time points. So this whole central dogma is very tightly regulated when it comes to differentiation. Unless until we get a differentiation of a particular DNA into RNA, say transcription regulation, we won't see the protein production. And once there is a protein production, then we can see the functionality or the functional aspect of a protein in a proper way. The other important things are, apart from the transcription regulation, there are cellular environment which are associated with the cell fate. Now cellular environment is also multifaceted, which we'll look into that in gradually, that what are the things which are responsible for making us fate, and the environmental could be related to the cellular environment. This is not the environment of a climate, but these are the environment of the cellular system. For example, the mechanical stress is there, some kind of a cell-to-cell adhesion molecules, another kind of structure which are associated with cells, such as extracellular matrix are also there. So we'll see that how this cellular environmental factor can decide the fate differently. Now, since I said that these are very important transcription regulation, and one such very important transcription regulation pathway is called Notch signaling pathway. So this Notch protein is one such pathway. There are many other pathways such as WNT pathway, Hedgehog signaling pathway, and there are many more which decide the fate of a cell to be differentiated into a different cell type. Some are very active in embryonic stage, where a zygote can differentiate into a different cell type, and further different cell types are differentiated into organ and organ systems. Now this Notch signaling, let's try to look into the complexity of this Notch signaling to some extent, and this will give you an idea that how a protein, a single protein, can decide the fate of a cell population. So let's try to understand what is notch signaling. Now when it comes to signaling, it's a downstream cascade. So for example, there is a cell and there is something happening and which lead to the production of certain things through the signaling pathway, which is happening from the external stimuli, and then this can lead to the fate of different cell functionality. So there is a protein which we will discuss in this pathway. As I told, this is just a kind of an example pathway, but just to give you an idea that how critical one pathway could be. So notch proteins are very important evolutionary protein. These are transmembrane type 1 protein. When it comes to type of, suppose you have a membrane, there could be a protein which is passed. So this is a transmembrane domain, this is an N-terminus, and this is a terminal. So this kind of a structure is called type 1 membrane protein. So this is type 1 membrane protein where the N-terminus is outside, and then you have a small hydrophobic stretches which are crossing the plasma membrane, and then there is a C-terminal end which can downstream process the pathway signaling cascade later to the cells. So these are transmembrane receptor proteins. It acts as a cell surface communicators. So what do you mean by that? That there could be some molecules which are coming in contact with this, and once these molecules are coming in contact, if there is a cognate binding with these molecules with this receptor, then you see that there is an activation, and then this activation can lead to further downstream processing. So these are kind of a cell surface communicator. So these are present on the very outer side of the cell. As I told earlier, that this is very crucial for cell fate determination during the developmental stage, tissue repair, and differentiation. So we looked into differentiation, how differentiation is very critical, that one cell type can be converted into multiple cell types. Tissue repair is also very important. So for example, we know that liver is a highly regenerative organ. So if there is any damage in the liver tissue, the hepatocyte can be regenerated in a way to repair the whole system, because liver is an organ of detoxification and we need to repair the cell types, particularly when it comes to damage of the liver cell. There are diseases such as hepatitis, which could be a viral specific or could be by toxin or any other substances where you require to have some kind of a repair mechanism from the cell system. So these mechanisms are inbuilt, and cell repairing system is generally activated when there is a requirement. Any excessive uptake or any excessive activation of this cell type specific differentiation could lead to cancerous condition. So there could be a very tight regulation of this transcription factor, and therefore we need to understand this notch signaling very critically. This is just one example, of course. They facilitate, as I told, cell-to-cell signaling. So of course this molecule which are coming from outside could activate this, or otherwise there is another cell which is coming in contact with this receptor to start. So basically this is cell 1 and this is cell 2. So there could be cell-to-cell communication which decide the signaling pathway. The other thing very important is we know these are related to differentiation, but this differentiation can lead to multiple different type of processes, such as neural differentiation. So notch signaling is essential for determining the specialized cell type, particularly for example the neural differentiation, how the central nervous system, the astrocyte, glia cell, and other kind of cells are differentiated from a single mass is largely depend upon the notch-like signaling pathways. And as I said, since these are very critical, any deviation or any mutation in the notch signaling pathway or notch signaling related proteins can lead to disorders, and these disorders could be developmental defect, it could be related to blood cancer where the T cell functioning is not proper, or there could be a way where cardiovascular diseases could flare up, where the heart and the related things related to heart such as blood vessels and capillaries are not functioning properly because of non-differentiation of the cell types. So this is one such protein which is critical. Now let's see how this signaling pathway is happening using this notch protein. Very important protein when it comes to early developmental stages. So let's see this notch signaling. So we could see that this is the cell type where this we could call cell number 1, and this is the cell number 2, and cell number 2 is having some kind of a ligand called delta, for example, here. So delta is one such ligand. So delta is a ligand, and we could see here the notch protein. Now these notch proteins are, if you can see the structure wise, it's a highly complex protein. It looks like an heterodimer. So this is one such unit, and there is another unit coming from this side. So both are joined here, and there are a lot of epidermal growth factor like domains on the top which can activate and bind to the ligand from the outside. So these proteins are very complex protein. There are many other notch like isoforms available in human body, but we are just discussing a prototype here. So these epidermal like growth factor domains which are present in the notch extracellular domain can interact with the delta ligand, and once there is a binding, we can see that there could be one proteases such as A disintegrine and metalloproteinase, also called ADAM10, for example, can do a cleavage here. And once there is a cleavage, then we can see that one of the fragment is dissociating into the cytoplasmic domain, and the N-terminal domain, since we are talking about a type 1 membrane protein, can cleave this one, so that this can bind to the upper domain of the notch, can bind to the delta ligand, and then this could be an extracellular domain can bind to this, and it can be go through the receptor endocytosis into the second cell type. However, what is happening to the cell 1, which is a critical cell? Now this detached a part of the notch can further cleaved into multiple different proteases, and one such is quoted here called gamma secretase, which is another protease is available in the cell, can cleave it into this called NICD, which stands for notch intracellular domain. And one could understand that once it is inside the cell, you call it intracellular, and when it is outside, you call it extracellular domain. So extracellular domain is gone along with the ligand, which is delta, and the intracellular domain notch can penetrate, since it's a transcription factor, and this transcription factor has to go to nucleus in order to start the processes, and therefore through nuclear pore complex these can migrate to the nucleus, and once it migrates to a nucleus, it go and attacks to the target site. So there could be a co-activators where the target can be bind, and there are some genes which are reported for notch signaling, such as MAM, which is called transcriptional co-activator. Name goes with a very specifically mastermind, but these are transcription co-activators. And then we have a CSL, which is a core DNA binding transcription factor. So once these complex can bind to the transcription promoter region or transcription regulatory region, this NICD, once coming in contact with this, can activate the target gene transcription. And once there is a target gene transcription, we see this particular cell type is destined to do some differentiation, some cell type differentiation, and this can lead to a whole bunch of different cell lineages. Now there could be possibility that this notch may not bind here, and there could be a complete shutdown of the transcription process. And if there is a complete shutdown, we see that because of the no notch signaling, or there is no notch protein available here, this pathway is blocked, and if this is blocked, there is no transcription, and we won't see any sort of cell differentiation. Now, as I said, that this is one such example, very complex pathway, and we can see that there is a protein on the cell surface. Once it binds to the ligand, it can do a whole bunch of cascade downstream, leading to a transcription of a gene, which can lead to a whole bunch of different subset or the sets of the cell which are required for the functionality of a cell. And if there is a deviation in any of these, we can see that you won't get a differentiated cell mass, and these are such a critical thing that can control the differentiation cell in the embryonic stage, as well as different stages of the tissue repair, or kind of a damaged tissue cell type can be differentiated by or repaired by this all processes. So let's move on. Since we understood the signaling pathway, let's try to see a different way. As I said, that apart from the notch signaling, we have some environmental factors which are restricting the cell fate, and we could discuss it in a detail. For example, matrix stiffness. So once the cells are always there in the extracellular matrix environment, how this cell lineages are supported by this matrix environment decide the cell fate. For example, the matrix stiffness could be a very low or rigid, a very dense. In that case, probably we can see the bone which are very tough in nature, while there could be a cartilage condition where the bones are cartilages are very flexible in nature. So the matrix stiffness decide the cell fates as well. There could be multiple examples of it. The second is topography or the cell's shape. So again, the environment, the physiological extracellular matrix environment of a cell type can decide whether it is an elongated cell. For example, the neurons, which are kind of very elongated cell mass, or the cell types such as RBC or WBC, which are spherical in nature. Then we have a myocyte, which is tapered and elongated cell types. Each cell types are destined to do multiple functions, and the fate of this can decide based on the cell type, shape and size and the topography of those cell types. The second thing, the other thing is mechanical forces. So when we see that the human bodies are always under the stress condition of multiple external forces, so when there is a stress in terms of forces, mechanical forces, we can see that the cell fates are decided on that, and there could be multiple proteins which are getting up or down regulated. Also there are channel protein which are getting opened or closed upon this mechanical stress, and we can just quote one such example in the next slide, where the protein can be opened upon the mechanical stress. So most of the cell-to-cell junction protein, for example, the tight junction, gap junction, these are the proteins which are responsible for making this junction proper functioning. So this mechanical stress which are there in the cell-to-cell junction can decide the fate of the cell population. So this is just an example of mechanical stress. So we can say that there is a mechanical stress happening in all the time, and this is just an example. So there is a protein called piezoic protein which is responsible for a cation based channel specific moiety that depends upon the mechanosensing. So say this is an extracellular space and this is cytoplasm, and these are the channel protein called piezo channel. There could be 1, 2 piezo different proteins are there. So we are not going in detail of that, but just for example that these are closed and there are cations which are outside in the extracellular space. Cell may need those cations or may not need those cations, but once the cations are required, then there could be a possibility that these piezo channels can open, and this opening can lead to the internalization of these cations into the cellular system. So it has been shown that when there is a mechanical force in the cell membrane, we could see that these ion channels are opened, and once these are open, they can give entry to this cation into the cellular mass. This could be a way the cells maintain the homeostasis, or this could be a way to maintain the homeostasis balance of the ion from outside to inside, because if there is a stress or the forces coming out because of certain thing, then we can see that some of the ions are penetrating inside. There are multiple studies which showed that the mechanosensation of these protein are responsible for a variety of cellular fate, including cancer. So these proteins are just one example how the cells can sense the stress, particularly the force. So let's move on, and we will see some other environmental cues which restrict the cell fates. So the next one is cell-to-cell interaction. So we know that once the cells are coming in contact with the other cell with the help of a receptor, we could see that the receptor cell interaction can lead to a variety of signaling cascade. We just have seen how notch protein can regulate the cell to cell interaction in terms of transcription regulation and other things. So that interaction of the cell to cell is also decide how the cellular fates are. In cancer, biology, we called it contact inhibition phenomena. And what does it? It stands that once the cancer cells, which are growing uncontrolled way, comes in contact with two cell type, their growth or mitosis get stopped there, because there is a contact inhibition. Now there could be a lot of regulation that might be happening that can lead to this phenomena, but once those two cells are coming in contact, then the cellular mitosis gets stopped right there, and this is what you call contact inhibition. And say each cell type of in the human body has certain lengthwise in terms of how many times it can divide. So each cell type can divide to certain extent. It cannot be divided uncontrollable, otherwise it may lead to cancer. So there could be a Hayflick limit, which people used to call what particular once the cell is dividing into say 4 times, 5 times or 6 times, for example. It could call that the Hayflick limit of that particular cell type is like 6 or 7. So the cell to cell junctioning is very important, interaction is very important, ligand based interaction or receptor based interaction. The fifth one is soluble factors. We know that there are a lot of cytokines which are secreted upon the cell-to-cell interaction. We have looked into the exosomes which are coming from one cell to the other cell and communicating in a different way. We have few idea about few of the cytokines, and we largely trying to understand how these cytokines can regulate the cellular fate. We have looked at some of the cytokines such as interleukin-2, which is called mitogen, or specific biological mitogen, can help differentiating the B cell lineages, which can lead to antibody mediated immune response, or in a proper way very categorically can raise the immune response in a proper way. So these are some of the soluble factor or the cytokine which can decide the cell fates. And finally, the amount of nutrients which are available to the cell mass. For example, if the nutrients are more, the cell can be hypertrophic in nature. Say when we see this, suppose this is a cell, when there is a more nutrition, the cell become hypertrophy. It doesn't mean that this is cancerous. This is simple in hypertrophy, which got hyper means more, trophy means nutrition. So if a particular cell is getting a more nutrition, we can see the size of the cell is increased. And then we could also see that the cell fat can be decided based on oxygen availability. Say whether the condition is hypoxia, which is less of oxygen, or there is an optimum amount of oxygen, or there could be some other level of oxygen. So these all nutrition and the oxygen level in a cellular mass can decide the fate of the cell differently, because oxygen can decide the various level of activities inside the host cell, and also the nutrient, for example glucose. If the level of glucose is more, we can see regulation of glycolysis differently, and which in turn lead to the production of ATP by mitochondria differently. So the nutrition can indirectly give the fate to the cell, because you see a different gradient of ATP generation, a different gradient of glycolysis, and so forth, and similarly for oxygen as well. So these are some of the environmental cues which are restricting the cell fate, apart from the transcription regulation by the signaling pathway, such as not signaling pathway. So these are all the way how the cells are differentiated into a multiple different lineages, and we'll see the plasticity of this cell type and its regulation later. But let's try to see some of the things which are related for early embryonic development and the later phase. I'll just give an example of the macrophages. For example, we'll see that macrophages are important antigen presenting cells, and these are all done in a different stages in the life cycle. For example, if you see here the prenatal origin, prenatal stands for just before birth. So embryo is there, and embryo can do multiple cell type in a different sense. For example, in the fetal liver, the monocyte can be producing the macrophages, and also yolk sac can produce a monocyte. So we could have a different set of the macrophages either coming from the liver or from the yolk sac. While in the later in the life, say after birth, we see that the different lineages of the macrophages are coming from the bone marrow. Say the bone marrow can convert into hematopoietic stem cell, which is common myeloid progenitor cell, and further it can convert into monocyte, which we have seen in the first slide as well. So say the development of macrophages, apart from the signaling pathway and all, also different during the different phases of life. Their differentiation and the origin is different in the prenatal, just before birth, and after birth, postnatal also their differentiations are quite different. Now also the macrophages could be very different when it comes to tissue level. So we can see that the cells are different in the bone marrow, different in the yolk sac, different in liver, but when it comes to the different lineages which are coming in the tissue space, now the function of these macrophages are simply engulfing the foreign pathogen or foreign substances coming to the body system. So there are cells which are basically called tissue specific macrophages, and we will go one by one in many instances when we will discuss the fate of this cell type in immune response. For example, in bone, we understood that the bones are made by the osteoblast, which are the mother cell, but then there are cells called osteoclast, which are responsible for the reabsorption of the bone cells. So there is any kind of a death, debris and all that can be engulfed by the osteoclast cell. So these are very specific macrophages, differentiation in the bone. Similarly, in central nervous system, we have a microglia cell which acts as an antigen presenting cell there, and also these are different differentiation of macrophages in a different way. In the skin, we see Langerhans cells, which are again cells present in the skin to engulf or trap any of the things which are coming in the body surface. Kupffer cells are very specifically for liver, and these are responsible for, these are present in liver sinusoids, an area of the hepatocytes randomly, which can help to trap the pathogen or the entering antigen into the liver system. Alveolar macrophages are again very important cells in the lung, which can trap the foreign dust particle or anything which are coming and giving harm to the lung functionality. So these are very important macrophages which are tissue specific and doing their function, while there are other macrophages which are very specifically present on the tissue level. For example, in the red pulp of the spleen, we see a different set of macrophages which can do a lot of function, because the spleen is graveyard of RBC, and post destruction of RBC, RBC2 has to renew itself for the salvage pathway to produce multiple other things. And then we have an area called marginal zone in the spleen, where we see a different set of macrophages. And of course, there are metallophilic macrophages which can engulf some of the metal. For example, if there is a destruction of RBC, we need someone who can engulf the iron, or particularly the other cofactors such as manganese or magnesium which are present in the system, so that the things can be recycled in a proper way. So we could understand that the cell differentiation is very complex, and each cell type are destined to do certain function in the human body. And again, these are controlled very critically with the transcription regulation, and also by lot of environmental factor which can decide the cell fate. And of course, we have gone to some extent in order to understand this, but still we need to learn how this plasticity of the cells are maintained, and what are the factors which can cause this plasticity stuff to happen in our next class. So we'll see the details of other processes in this lecture in our next class. Thank you.